would like to invite our last speaker of the session, of today's session, Mr. Rahul Kumar. Sir, please unmute yourself.
Yes, ma'am.
Okay. So Mr. Rahul Kumar is working as Assistant Professor and Head, Department of Zoology, Theodenic South College, Kaler, Arwal, Bihar, which is a constituent college of Magadh University, Bodh Gaya. He has also worked in many other renowned academic institutions of India like Jawaharlal Nehru University, New Delhi, National Council of Educational Research and Training, New Delhi, Indian Agricultural Research Institute, New Delhi, and All India Institute of Medical Sciences, New Delhi, as a research scholar. At AIIMS, New Delhi, he has worked in a DST nanomission funded project. He has research experience in variety of fields like inclusive science education, activity-based teaching learning methods, molecular biology, biotechnology, functional genomics, nanotechnology, molecular taxonomy, etc. He has few scientific publications too. He loves also teaching, exploring new things, and believes in not being limited to concrete labs. Thank you, sir. The title of his talk is An Efficient Nanoformulation for Ophthalmic Application. Sir, please proceed.
Thank you, ma'am. Respected organizers and eminent scholars, as I have already been introduced, my name is Rahul. And today I am going to briefly present a part of my research work, which was conducted at AIIMS, New Delhi. Although right now COVID-19 is in limelight, but India holds a major share of population suffering from many other infectious and non-infectious diseases globally. So the title of my presentation is An Efficient Nanoformulation for Ophthalmic Application. We have named it Nanobrimonidine. I will be discussing this topic with respect to a disease named glaucoma. And also, I have tried to keep my presentation student friendly because many of the participants, the listeners are also students. So in various slides, there may come some pictures of instruments and techniques. So it is especially for students. So glaucoma, it is also called Kala Motiyabind in Hindi, and India accounts for approximately 25% of global glaucoma related blindness. In India, glaucoma is the leading cause of irreversible blindness, with at least 12 million people affected and nearly 1.2 million people blind from the disease. Glaucoma is a group of eye diseases. Actually, there are multiple types of glaucoma, and a specific type named open angle glaucoma. It is the most major type of glaucoma, and it is found in wide number of cases, and it can cause vision loss and blindness by damaging a nerve in the back of our eye called the optic nerve, which is present just in the middle of retina behind the blind spot. That is called fovea centralis. This damage is often caused by an abnormally high pressure in our eye, as I will explain in the next slide. There is no cure for glaucoma, but early treatment can often stop the damage and protect our vision. On the right side, you can see the photographs. The upper panel shows a person suffering from cataract, and the lower panel shows the eyes of person suffering from glaucoma. Many people confuse between cataract and glaucoma because in Hindi both are called motiyabind, but cataract is simply motiyabind or white cataract, we can say, because a cloudy, hazy kind of formations or encrustations, they occur over the lens and just beneath the cornea, and it can be removed by operation. But in glaucoma, the symptoms cannot be visualized from outside. That's why glaucoma may proceed to advanced stages and remain undetected for a long time. Now, I would like to explain the anatomy of human eye with respect to open angle glaucoma, because most of the listeners and participants are from physics background. They must have studied about the anatomy of eyes in optics, but still I would like to explain the anatomy of human eye with respect to glaucoma. So as you can see, the the eye consists of two chambers, one posterior chamber and one anterior chamber. The posterior chamber is filled with vitreous humor, and the anterior chamber is filled with a fluid called aqueous humor. Aqueous humor is watery fluid, and vitreous humor is viscous. It is gel kind of thing. And what happens that this aqueous humor, these keeps on getting produced in our eyes, and they need to be drained out through a structure called canal of Schlemm. As I am showing with pointer, this is canal of Schlemm, and this canal of Schlemm is guarded by a specific tissue called trabecular meshwork. The aqueous humor gets filtered through the trabecular meshwork, and it comes out of the eyes consistently, and it keeps our eyes moist. But what happens in glaucoma, that due to some reasons, for example, excess tissue growth, inflammation, or limited blood supply, this trabecular meshwork gets obstructed, due to which the aqueous humor is not able to come out of the eyes, due to which it keeps on building inside the anterior chamber of the eyes. The consistent building of this fluid leads to increase in intraocular pressure, and when this pressure increases, it damages the optic nerve, and if it remains untreated, this may also lead to permanent blindness. So Brimonidine is one of the clinically most recommended drugs for glaucoma. It is considered, in fact, the wonder drug for glaucoma, and it acts upon the trabecular meshwork of eyes and reduces intraocular pressure by increasing aqueous outflow. So how does it increases aqueous outflow? This drug, it can create pores in the trabecular meshwork. But it shows poor bioavailability, because most of the drugs associated with ophthalmic diseases are given in the form of eye drops. So what happens that most of the Brimonidine gets lost in the eye drops, due to which higher doses are recommended. But higher doses have several side effects, such as itching, blurring of vision, puffy eyes, nausea, irregular heartbeats, shallow breathing, and numbness in hands and feet. So there is a solution, as observed in case of many drug formulations. Ultra-small chitosan nanoparticles work as an ideal drug delivery vehicle for many reasons. But the major problem associated with chitosan nanoparticles is the successful synthesis of ultra-small sized nanoparticles, which is less than 50 nanometer, which show better penetration through the cornea to reach the posterior segment of the eye. Therefore, we have reported the nanoencapsulation of Brimonidine on ultra-small chitosan nanoparticles, which we have named Nanobrimonidine, by a co-synthesis method based on modified ionotropic gelation process, which has been developed by our lab previously, and this process has also been patented. Before moving further, I would like to give an introduction to the chitosan. Previous speakers, they have told about cancer and application of other nanoparticles in treatment of cancer and other aspects. But as Tapas sir mentioned, the nanoparticle he used was considerably toxic, as he said, and it should be removed from the system gradually. But chitosan is non-toxic. It is biodegradable. It is a polysaccharide derived from chitin, and chitin is second most abundant polysaccharide in the world after cellulose. As you can see on the right photograph, I have added a few photographs of these are crustaceans. These are arthropods like prawn, which we call jhinga machli in Bihar and North India, and other like crab and lobsters. And many other arthropods, they contain chitin in their outside shell or exoskeleton. Chitin is also found in the cell wall of fungi. Chitosan is produced by alkaline deacetylation of chitin. The process of alkaline deacetylation, it is industrially performed, which I will be explaining in the next slide. Chitosan is biodegradable, as I already explained. It is biocompatible, polycationic, naturally derived polymer with low immunogenicity. It also has antimicrobial properties, therefore it can be used in medical applications. The process which I have used for the synthesis of chitosan nanoparticles from chitosan polymer is ionotropic gelation. As already explained by Tapas sir, he explained two types of method of nanoparticle synthesis. One is top-down approach and one is bottoms-up approach. So the approach which I have used, which is inotropic gelation method, is a kind of top-down approach. It is a technique that allows the production of nanoparticles and microparticles by electrostatic interactions between two ionic species under certain conditions. At least one of species has to be polymer. In this case, chitosan is the polymer. Now, this is the process by which industries synthesize chitosan from chitin. These are different different steps of synthesis of chitosan. First, the living tissue is grinded, then it is decalcified, because most of the crustaceans, they are marine, and the seawater is rich in calcium salt, so there is abundance of calcium in their shells. So decalcification is occurred either by treating with dilute aqueous HCl solution, and then deproteination occurs by the use of protease producing bacteria. Proteases are enzymes which can degrade proteins. Then decolorization is occurred, which is followed by deacetylation. Deacetylation can be done using chitin deacetylase enzyme or hot concentrated NaOH solution. Both can cause deacetylation to produce chitosan polymer. So this is the process how industries produce chitosan. Now I would like to present a brief account. How did we produce chitosan nanoparticles? Actually, various parameters have been standardized and it is patented. I am giving a brief overview. As I already explained, it has been synthesized using inotropic gelation method. So we have used one ionic particle, one ionic polymer as chitosan, which is positively charged, and it is dissolved in acetic acid, and we use another polymer, sodium tripolyphosphate, which has 3 phosphate groups, which is negatively charged. So the final formation of the nanoparticle is the result of electron interaction between these two ionic particles, and we mixed Brimonidine with sodium triphosphate solution only, tripolyphosphate solution only. And these were added to the chitosan solution dropwise. Even the speed of drop matters for the synthesis of ultra-small chitosan nanoparticles. We have kept the speed of drop as 1 drop per second. So after on the right panel, as I already said, that I will be also in most of the slides, I will be sharing the diagrams and photographs of the instruments which I have used during my work for the purpose of knowledge of students. So on the right side, you can see this is a bath sonicator. It is used for the homogenization and breaking of the chitosan polymer into its constituent pieces, which is an important step in the top-down approach of synthesis of nanoparticles. So after synthesis of nanobrimonidine, its characterization was done. So there are different properties which are kept into mind before characterizing a nanoparticle. A nanoparticle has a size. It, as the name suggests, nano comes before particle. So it has dimension in the range of nanometer. So first of all, its size is important. Second, its charge is important. So to measure particle size and particle charge, there is the instrument called Zeta particle size analyzer, which you can see on the left panel. And it works on the principle of DLS, that is dynamic light scattering. And after size and charge, another important thing is its structure, its morphology. How does it look? So for that purpose, we use electron microscope. There are two kinds of electron microscope, transmission and scanning electron microscope. In the middle panel, you can see cryo-transmission electron microscope. It has recently been installed at AIIMS, and it is an upgraded version of transmission electron microscope, which can also be studied for structural studies of biomolecules like proteins, RNA, DNA, etc. And after size, morphology, charge, and structure, another parameter is its concentration. Sometimes what happens in a solution of nanoparticles, there may present more than one entities. So for that purpose, a spectroscope identify species and its concentration. So I show you visible photometer. So explaining these things, I would like to proceed with my data. These are the data I have characterized the ultra-small chitosan nanoparticles, Nanobrimonidine, and PF68 capped Nanobrimonidine. So what is PF68? PF68 acts as a stabilizer. It works like a surfactant. It caps the Nanobrimonidine particles in the solution, and what it does, it prevents aggregation of Nanobrimonidine over time. We also analyzed the stability of Nanobrimonidine after 1 month storage without freeze, one month post time. And you can see in conditions the size and charge of the uh, nanobrimonidine, even the morphology of the nanobrimonidine was not altered significantly. It remained constant over a long time. When it comes to the matter of charge, I would like to tell one thing, that the charge should be cationic. Why it should be cationic? Because the purpose of using this nano formulation is to increase the availability of the drug. The drug availability can only be increased when this
तीन हो गए।
Hello, sir.
Yes, ma'am. There has been some internet-related problem. I am
Okay,
again
sir.
sharing my screen.
Okay, sir.
Okay, am I audible now?
Yes, sir, you are audible.
Okay, sorry for interruption due to some network problem. It happened. Okay.
Okay, sir, no problem.
I have already explained this slide. Was it visible when I was explaining it? Okay.
Yes, sir, it was visible.
Okay. Okay.
TEM instrument sites was visible.
Okay. So I characterized the nanoparticles under different conditions, and I found it consistent with respect to size, morphology, as well as stability. So why charge matters? The chitosan nanoparticles must be cationic. They should be positively charged because the cell surface is negatively charged. Every cell of our body is negatively charged from outside. So if it is positively charged, then it can stick to the cells and it can keep releasing the drug consistently, therefore reducing the loss of the drug. So next, we also characterized our drug using UV spectroscopic methods like UV visible spectrophotometry and Fdir spectroscopy. In both, you can see there are two peaks. These two peaks are characteristic peaks of Brimonidine. One is at around 240 nanometer and one is around 320 nanometers. The maroon peak, it is of only Brimonidine, which we used as positive control. The yellow peak, it is after purifying the nanobrimonidine using dialysis in HPLC. And the orange one is the nanobrimonidine using chitosan nanoparticle as blank. And the green one is only chitosan nanoparticle. So it is confirmation of the nano encapsulation process. To further confirm the process of nano encapsulation, we performed Fdir spectroscopy. Fdir stands for Fourier transformation infrared spectrum spectroscopy, and Fourier transformation is a mathematical algorithm through which the it is characteristic of Fdir spectrum is characteristic of every molecule, and by analyzing the molecular profile, it provides different peaks. So you can see on the left graph, it is just for chitosan, and in the middle one, this contains only Brimonidine. And on the extreme right, you can see the peaks for nanobrimonidine. It consists of peaks of both chitosan as well as Brimonidine. So it confirms our nano encapsulation process. After confirmation of nano encapsulation process, we also determined the concentration of Brimonidine. Actually, we knew the concentration of Brimonidine, but we confirmed it by plotting the standard calibration curve using different concentration of Brimonidine of the known concentration. And we got 200 microgram per ml. It was the initial concentration used, and it was 67 microgram per ml. It was in the reaction mixture. It is the final concentration. And the encapsulation efficiency based on this calibration curve data was calculated to be 40%, which is quite convincing for such a kind of drug. And we also checked after performing all these experiments, we wanted to see the activity of nanobrimonidine, whether the purpose of making nanobrimonidine has been successful or not. So we performed in vitro release assay of nanobrimonidine using dialysis membrane and isotonic solution inside the dialysis membrane, dialysis bag, and outside dialysis bag. And we found that within half an hour, the burst release of the brimonidine began, and burst release should be there. In pharmacokinetics, every drug should initiate burst release in order to begin the process, and it should consistently keep on doing its functions only if it is present at the site of the action. So you can see that within half an hour burst release started, and about half of the brimonidine was released from our nanoformulation by 45 hours, and by within 100 hours, 100% of the nanobrimonidine was released from the nanobrimonidine formulation, which is very convincing. And after doing in vitro release assay, we also checked the cytotoxicity of nanobrimonidine using L292 cells. These are cultured lens cells, and we performed this experiment in cell culture using an experiment called MTT assay. So we found that even at a concentration as high as 1 gram per ml, the cell viability was not reduced below 80%. So it was quite good for this formulation. Further, we also wanted to check the in vitro uptake mechanism and time-dependent uptake of nanobrimonidine by L292 cells. So to perform in vitro uptake mechanism, to check in vitro uptake mechanism, we treated the cultured cells with different inhibitors of endocytosis. Especially for students, endocytosis is the process by which a cell takes into foreign materials, internalize the foreign material which is present outside the cell. So there are different types of endocytosis like clathrin-mediated endocytosis, phagocytosis, pinocytosis. So we found that when we inhibited the cells with indomethacin, the internalization of the nanoparticle was highly reduced. It confirms that the nanobrimonidine is internalized by L292 cells via caveoli-mediated endocytosis pathway, which is a kind of receptor-mediated endocytosis. And this is the kind of endocytosis which is inhibited by indomethacin. After performing this assay, we also performed time-dependent uptake of nanobrimonidine, which you can see in the right panel. And we found that within 4 hours, nanobrimonidine was able to be uptaken by the cells in the amount as high as 80% without showing any toxicity. So it serves our purpose. Finally, we wanted to confirm, because what happens many times that after a nanoencapsulation process or after conjugation of a drug with nanoparticles, the functionality of the drug and nanoparticle gets lost. So to confirm whether our nanoformulation is functional or not, we performed first assay. I told previously that chitosan nanoparticles are positively charged. That's why they show mucosa adhesive nature. So wanted to check whether nanobrimonidine is also mucosa adhesive or not. So we treated bull eye cornea with rhodamine labeled nanobrimonidine. Rhodamine is a fluorescent dye. It is red colored dye. And we labeled nanobrimonidine with rhodamine, and we treated bull's eye cornea. And after treating, we left it for 1 hour and we washed the bull eye cornea. And we found that even after washing, the fluorescence was visible under fluorescent microscope at 80% intensity. So this is one of the functional characterization of the nanobrimonidine. Finally, to check whether the brimonidine present in nanobrimonidine is able to function at par with free brimonidine. So we treated a trabecular meshwork tissue. These tissues have been obtained from the eyes of glaucoma patients, and I brought these tissues from Rajendra Prasad Institute of Ophthalmic Sciences, AIIMS, by the help of Dr. Tanuj. He is professor there and a renowned ophthalmologist of India. And what we did as a negative control, we treated these tissues with empty chitosan nanoparticles without the drug. We also treated the tissue with free brimonidine as well as nanobrimonidine. You can see as shown by arrows in each of the micrograph, you can see in both in case of free brimonidine as well as nanobrimonidine, the pore formation was visible. You can see the right panel is showing the scanning electron micrograph, and the right panel is showing transmission electron micrograph. In both of the cases, you can see formation of pores. So this serves our purpose, and it confirms the functionality of brimonidine present in the nanobrimonidine. So this was the last data I had to share. Now I would like to conclude. Nanobrimonidine has the potential to be used for open angle glaucoma due to its ultra small size, that is 28 nanometer, higher drug loading capacity, stability, longer controlled release, and better functions on dilation of the trabecular meshwork. We have developed a new and simple method for the synthesis of monodisperse ultra small nanobrimonidine using modified inotropic gelation between low molecular weight deacetylated chitosan and STPP, as I already explained. The average zeta potential of nanobrimonidine is plus 37 electron volt, which is must for its mucosa adhesive property. The particles were stable for months after freeze drying, and in vitro release study revealed that the burst release occurred in half hour and complete release in 100 hours, which is needed for the consistent release. These particles showed no cytotoxicity. The internalization of nanobrimonidine showed the maximum uptake through receptor-mediated endocytosis pathway, which we can say more specifically as caveoli-mediated endocytosis. The developed nanobrimonidine has good potential for the dilation of trabeculectomy tissue of the glaucoma patient, as I have already shown in the last data. So what's next? This entire experiment has been done in the in vitro condition. It can only become fruitful if it is done in vivo condition. Only after that it can be used on human beings. These are the fundamental steps of drug development. So in vivo trials are still pending, and experiments on live animals are still going on in our lab. So lastly, I would like to acknowledge all my three mentors, Dr. Indu Barwal, who is right now scientist at the Regional Center for Biotechnology, Dr. Tanuj, who is professor and ophthalmologist at Rajendra Prasad Center for Ophthalmic Sciences, AIIMS, my supervisor, Dr. Subhash Yadav, who is working in Department of Anatomy, AIIMS, and my lab mates for their support in AIIMS New Delhi for funding this project intramurally. So thank you. This is the publication from my work, which has recently been published by our lab in Microscopy Society of America official journal, Microscopy and Microanalysis. Thank you, and questions are welcome.